For the technical difficulties. First seminar at the BIMR. I'd like to welcome you all. My name is Alan Sadrov. I have the distinct pleasure of directing the Brockwell Institute for Materials Research, which hosts this seminar series. And this is the very first of the seminars for the year. And I'm extremely pleased that we have a really great seminar for you today. And our speaker will be introduced by Sam Sadeghi, who is the organizer of the theme on sort of radiopharmaceuticals, which we will have a series of 4 lectures about. So the next 4 lectures are going to be in this theme, and I hope that you will come back for the remaining 3 after today. So with that, Sam, I turn the floor over to you.
Yeah, it really is a privilege to have Neil here. You guys all probably know Neil by reputation. He is definitely the pride and joy of McMaster. I would even go further to say pride and joy of Canada in the field of molecular imaging. And alumni, he did his undergrad here, his PhD here with Schrödinger and Chiricol.
Yeah.
And after that, he went to the national lab in the US with an NSERC postdoc scholarship. And these are tough to get because I tried and didn't get one. And he was recruited right away to U of T to CAMH, to a faculty member there. He was recruited again by Harvard and NGH to run their radiochemistry, built that up. After a while, kind of was like, we want Neil back. He was gracious enough to come back, and he is back leading the CAMH neuro program, and he's overseen over the last few years a huge expansion. And he's gonna, I can talk about that, probably the biggest PET brain imaging center in the world. In fact, Neil, I would say, and his team and the CAMH team are probably synonymous with the best in the world when it comes to PET imaging of the brain, clinical trials. He's top notch, essentially. And maybe one quick little anecdote that he shared with me today. I think he's comfortable with me saying that. He was just telling that to everybody, so I'm guessing it's okay. We were just taking him to talk to the VPR. His comeback as a celebrity, of course, as a previous alumni. And he was at the Gilmore Hall, and he just told me the last time he was there, he was actually because he almost flunked Adam McMaster in his first year study. So here he is, a celebrated scientist, and I just want to let the students know if you do encounter any difficulties or setbacks, which happen, you will at some point. Neil is a testament as to how far tenacity and dedication he's taking. So with that, Neil, please.
Thank you for the super kind introduction. Thank you for the super kind introduction and roast, Sam. But he is correct. I do have those memories of McMaster there. It's actually surreal and nostalgic to even be here today. And I really did hate this room. ABB 102 is where I had computer science, and that was my lowest mark. That was my D-, and it ruined my cumulative average all the way through. So it's wild to be giving a lecture in the same room now after almost 30, it's over 30 years. So it's also a real massive honor to be speaking here too, because I started in '93, and in '94, Professor Brockhaus won the Nobel Prize. So just seeing this whole thing and giving a lecture in the Brockhaus seminar series is a massive honor. So again, thank you all for the kind invitation. As Sam mentioned, I'll talk about PET neuroimaging in drug discovery, and I want to show some applications of how my lab works with the pharmaceutical industry and how we do projects that have industrial interest. And I mentioned we'll be talking about PET imaging. I know everybody's very familiar with MRI and CT scanning, where a patient lies in a scanner and you can get the structure of the brain. Or in neuroimaging, we really rely on PET for function. That's where we take a radionuclide, inject it in the body, and we can see how it distributes in the brain. Now, the way we make PET isotopes or radionuclides is using a particle accelerator called a cyclotron. The cyclotron that we use for medical imaging is quite small, not like the massive ones they have in national labs in the US. And actually, interestingly, the cyclotron was, for those of you who saw Oppenheimer, is a particle accelerator that he won the Nobel Prize for, which basically spins particles, and I can show how that works shortly. So I like to think of it, though, as modern day alchemy. I like to think of cyclotrons as doing the work of taking, like the 15th century chemists, trying to turn base metals into gold. We use a cyclotron which takes a particle like a proton, splits it to become H+, And it'll go onto these semicircular copper electrodes shaped like the letter D. And we switch polarity at 24 million times per second, so 24 megahertz. The proton that's positively charged goes to the negative charge, and it's constantly chasing the negative charge as we switch polarity. In a magnetic field and under vacuum, it starts to spiral outward and accelerate. Now, I know Troy and other physicists here, they hate when I do this, but it's 450 million miles per hour. But they don't like speed, but if we, it's 2/3 the speed of light and 10 to 17 mega electron volts. Once we have this super high energy proton beam, we hit it onto a target, and that's where the actual nuclear reaction takes place. We load it with a stable isotope, such as oxygen-18 in the form of water, hit it with the high-speed protons so hard that it bumps out a neutron and turns it into fluorine. So oxygen becomes fluorine. I've been doing this now for over almost 30 years, and it still blows my mind that this is the type of thing that we do in our lab every day. We do this also for with nitrogen, just in a gas target with nitrogen, just like the gas we breathe, but high purity, hit it with a proton so hard that alpha or helium comes out and it becomes radioactive carbon-11. These are very short-lived radionuclides, ranging from 2 hours down to 2 minutes. So you can imagine that the radiochemistry has to be really done almost at lightning speed. We're always racing the clock. Now, I mentioned PET employs specific molecules labeled with a positron-emitting radionuclide. This is an example of taking the most common radio tracer. It's called FDG, or fluorodeoxyglucose. It's a radioactive sugar analog, and it goes where sugar would go once it's injected in the body. For the chemists in the room, it's very simple. It's just an SN2 reaction and a deprotection. Historically, though, this was not easy to make. When I learned to make it here by my PhD co-supervisor, Raman Chirikal, it was made by hand using manipulator arms, and every lab in the world made it differently. They used different precursors, different fluorinating agents, different protecting groups. And we also had superstitions around it. In our lab, we said it won't work unless you're playing Van Halen. So it was literally that much of an art, but now it's down to a science where it can be done routinely with those cassette-based systems and shipped around the world, around the country. So this is an FDG scan of a normal individual about 60 minutes after injection, and you see radioactivity where you would expect it, the brain, heart, kidneys, and bladder. Applications in oncology are massive because you can imagine a baseline study where all those other black dots you see are lesions, pre and post chemotherapy. So this is an example where the chemotherapy really worked, and that's the goal. And you can use PET for something like that. So you can imagine the power of it. Our center is only focused on neuroimaging. Just to give you some background on the Center for Addiction and Mental Health, or CAMH, this was the 4th PET center in Canada at the time. When I started in the field, there was only 4 places one could train to do radiochemistry. There was McMaster, there was UBC, and there was McGill, and then the new kid on the block was this Toronto. We're in the heart of downtown Toronto and on the University of Toronto campus, but we have a really long history going back to 1850. Back then, it was called the Provincial Lunatic Asylum. And it had 12-foot walls around it built by the patients to keep them in and the community out. So you can imagine the stigma around mental health back then. But CAMH it was always been at the center of research, discovery, and innovation. So they invested in bringing down the stigma, integrating the center into the community, but also investing in a PET center. And our center has grown tremendously since. We have the first 2 cyclotrons in Toronto, 17 hot cells, which are the lead-shielded fume hoods, 2 labs, and everything to do bench to bedside development of radiopharmaceuticals, from chemistry, preclinical neuroscience, MRI, CT, human imaging, everything to go that way. We have about 100 faculty, fellows, staff, and students in our center alone. And we have, right now we're at 42 radiopharmaceuticals for human imaging. We have another 5 that will be submitted to Health Canada by the end of the year. So we'll be at 47 by the end of the year, and about half of those are bench to bedside, first in human use, and to study all facets of brain health. This is a snapshot of what we have. These are all the different tracers, pretty much all labeled with carbon-11 or fluorine-18. Purple is first in human, blue is first in Canada, and the rest are other. So you can see we try to image every type of receptor, enzyme, transporter, neurotarget that we can. And we really are trying our best to try and understand all facets of brain health, and even including brain cancers. In 2027, we'll move to what's called the Temerity Research and Discovery Center. This is $1 billion project, and they've taken all the input from the scientists. It's going to be shaped like a brain and an iconic building in the city. What's really cool about it for us is it's going to have 2 cyclotrons and 24 hot cells, making us the largest facility in the world that's dedicated just for brain imaging only for research. So we're really excited to move into this building in 2 years, and we think that'll bring us to the more cutting edge. Why are we investing so much money in brain imaging and building an imaging center? If you look at drug development, they start with about 1 million structures in pharma. And when you go through the pipeline, it takes 10 to 20 years and costs millions to billions of dollars. So anything you can do to de-risk drug development is worth gold to pharma. So you can imagine we use PET to determine the dose of a drug. That's critical because if the drug dose is too low, patients won't take it. It doesn't work. If it's too high, patients will get side effects. So we need to know the doses of drugs with PET. Another thing we do with pharma companies is we label the drug without changing the structure, and we inject it in the body and we see how it distributes. So we get classical pharmacokinetic data from that. We also confirm target engagement. So if we have a tracer that hits a specific target in the brain, we can test drugs and make sure they hit the target. And that's also worth a ton to the industry. But to do that, we need new radiochemistry methods and PET tracers to do that. We need to be really investing in that area. And that is a space where I feel like we're quite stagnant and we really need to expand on right now. Coming back to the different types of brain scanners, PET neuroimaging in Canada has a very long history, going back to the 70s at McMaster with a homemade PET scanner. By the time Toronto got involved in the 90s, they got this HRT, which is the highest resolution, had only brain PET scanner. UBC Triumph had built their own as well. When I started, I mentioned there was only 4 spots you could train. Now you can see PET has expanded all over the country. There's scanners everywhere that can be remotely shipped. You can remotely ship the tracers to. McMaster also had a very seminal role in understanding the living human brain for functional imaging, particularly in the dopamine pathway. So for those of you who've seen the movie Awakenings, it's a story where, it's a true story about Robin Williams and Robert De Niro play these physicians who take catatonic patients with extreme Parkinson's disease and cannot move. And what they did was you're trying to increase dopamine in the brain for a Parkinson's patient, but if you can't give dopamine itself because it doesn't cross the blood-brain barrier. So they started administering the metabolic precursor L-DOPA, which crosses the blood-brain barrier, gets enzymatically converted to dopamine, raising the levels and taking people who couldn't even move or walk to being able to walk around and actually function. And L-DOPA treatment is still used today in Parkinson's. The chemistry was always the biggest challenge though. It was almost one of the hardest things to do in chemistry is label a non-activated aromatic ring with fluorine. Fluorine is the most electronegative element. It's very challenging to work with it with organic compounds because it will basically set anything on fire, turn it into a tar, it'll react explosively. It'll pretty much tear apart any solvent as well. So there was some really fundamental work from the early 1980s led by my co-supervisors, Dr. Chirikal and Dr. Gary Strobeljan, who's here today. They came up with ways to tame fluorine by diluting it, using low temperatures, finding oxidatively resistant solvents like anhydrous hydrogen fluoride that don't get attacked by fluorine gas. This is an example, and this is a true story, and Professor Strobeljan was there with me when we did this. We blew out a fume hood. We were literally trying to make fluoride behave like F2. And I said, I'd never do it again after this, but I'm gonna come back to that point because that's still a holy grail in the field. Okay, so again, taming fluorine, getting fluorine 18 onto L-DOPA was a seminal moment. It was actually made, the dose was made by Professor Chirikal, and he injected the PET Center director at the time, so, Professor Steve Garnett. He got the cover page of Nature for his brain. And this is an example. You see a nice symmetric distribution of fluorodopamine in the striatum, where the majority of dopamine neurons are known to be present, and a huge reduction in Parkinson's disease. That's exactly what they were looking for. This is seminal for 2 reasons. One of them is it was radiochemistry that was unprecedented, putting fluorine 18 onto a non-activated aromatic ring. Still a holy grail today, and functional imaging being realized for the first time in PET, in a living human person. So this shaped the whole field of PET, drug discovery. I was fortunate to train in the glory days when you could make a PET tracer and inject your boss. So These three figures from my thesis are three tracers that we made that my PhD supervisor volunteered for. So unfortunately, you can't do that now because of the regulatory burdens, but it was a great time to be able to do science. Other labs were always like that too. This is an example of how one would use a dopamine imaging agent. This is a different tracer called raclopride in the brain. This is the striatum, and this is a patient on Haldol or haloperidol, which is an antipsychotic medication. And you can see the blockade from the drug. That means it's going to the striatum, the region of interest, and binding to the target. It's used still among one of the most widely used tracers worldwide. As Sam said, after I finished my PhD here, I did a postdoc at Berkeley National Laboratories, and I was looking for a faculty appointment. And the reason I was attracted to the University of Toronto and Center for Addiction and Mental Health is because they were also doing innovative chemistry and innovative imaging studies. They had come up with this really cool method. So 99% of carbon-11 chemistry is methylation. And they found that instead of just doing the reaction in a small vial, they did it right on the HPLC loop. And it's known as the loop method. It's such a simple method to put carbon-11 on molecules. So this was the heyday of the loop method. And then they were also figuring out the doses needed for antidepressant medications. Despite billions of doses of antidepressants, including Prozac and other SSRIs, nobody knew what dose to give. So what they did was use PET, and they found that 80% receptor occupancy was the sweet spot. So that's actually used today in clinical trials. And they were also adding this to using the same methodology for antipsychotics and other medications. Another thing I was very interested in is I had an opportunity to go there, and they said, you can work on anything you want if you come here. And I thought that was amazing. So that's why I started my faculty career there in 2004. And me and I think probably every other chemist in the room, I think they'd be lying if they said they weren't inspired by E.J. Corey and the logic of total synthesis. Basically, I was fortunate to meet him when I was at Harvard, and he joined me on one of my grants. But for the idea here is that E.J. Corey said 60 years ago, when he started as a professor at Harvard 60 years ago, people would say, you can't make this drug. So you can't make it. We don't have the methods. We don't have the reagents. So he said, we'll take the most complicated molecule, natural product, whatever it is, break it down to its simplest bits, and synthesize things in multi-steps, because we'll transform the field by coming up with new methods, reagents, and products along the way. So I feel in our field of radiochemistry, we're similarly stagnant. We're reliant on one method for fluorine-18 labeling, which is one-step fluorinations with fluoride and one-step methylations with carbon-11. So if we could label anything and chemistry wasn't a problem, we could envision total radiosynthesis. And now in our field, the goal is to label as late as possible, because these are short-lived radionuclides. But I think the harsh reality in our field is that most drugs cannot be radio-labeled for PET. But with total radiosynthesis, hopefully we'll be able to also come up with new radiochemical methods and reagents along the way. And we've shown that we can push carbon-11 out to 6 steps, and we can show that fluorine-18 can be pushed out to 10 steps. You don't have to just do one-step reactions. Another harsh reality is that most drugs fail, so it's not a surprise that most radio tracers also fail. So I feel if we should be making radio tracers that we actually want, rather than the ones we just can make, even if it's inefficient with multi-steps, and rapidly get them into preclinical models. If they work, we'll simplify the chemistry later. And you can use FDG as an example. Remember I mentioned every lab made it differently before. It was difficult to do. Now it's cassette-based, meaning we have a technician that comes in the morning, pops on a cassette, hits go on the laptop, sends the fluoride from the cyclotron, and FDG pops out in a vial. It's done all over the world and distributed and shipped now. So it's because when there's a need for something, we can figure out ways to make it viable for anybody. What I love to ask in an audience like this is, what if we could label anything, any molecule for any target? What would people be interested in, assuming radiochemistry is not a problem? So I asked this question in my first job at CAMH, and the answer I got was really interesting from the physicians. They said, we're imaging dopamine with fluorodopa. That's great, but that's a little tricky. We want to use it for drug development. And we're using compounds like raclopride that I showed, which are antagonists. But we want to know, because dopamine itself only binds the dopamine receptor when it's coupled to G protein in a high affinity state. So we want agonist radio tracers, because they also only bind the high affinity state. That'll be a more true measure, and we can use that to de-risk drug development. So we started looking in the literature, and every compound, nothing was easy to methylate, and none of them had fluorine on it. They all had N-propyl groups, which was a bit of a nightmare. But we were like, okay, maybe we can go, we'd start with carbon dioxide. Maybe we can just do this with organic chemistry. Even though it's a 20-minute half-life, we can make an acid chloride, react it with a secondary amine, reduce the carbonyl, and make N-propyl groups with carbon 11. And interestingly, we still use this method today. You see, it was translated to human. One of the analogs, you get such an intense uptake in the striatum compared to the other tracers. And it's just like total synthesis. The goal is to be concise, minimize the steps. And many, many labs have tried it, including ours, but there's no other way to do it for human use. So we still make it like this even today. So 6-step reactions can be used for human use. It doesn't have to be simple 1-step reactions. Second time I asked this question was when I started my second faculty career at Harvard Medical School and Mass General Hospital. We were making this compound called Pittsburgh Compound B, or PIB, which images amyloid plaques for the Harvard Aging Brain Study. So we were scanning people throughout their life to see if they would convert to Alzheimer's disease. So again, we had to make this tracer multiple times a day. This is a 74-year-old with low amyloid. This is a 72-year-old with Alzheimer's disease. If every PIB scan had that level of resolution, we'd be in business. But we're imaging amyloid plaques, which are a normal part of aging. And you get this huge cohort of people that they look the same. They have the same plaque load. And if we followed them even over 20 years, they don't even convert or necessarily convert to Alzheimer's disease. So we need more sensitive biomarkers. There's 3 FDA-approved radiopharmaceuticals for imaging amyloid plaques, but none of them are sensitive. So I bet my startup package on tau protein. And there was a company called Siemens Molecular Imaging Biomarkers at the time, and they showed this lead called T807. It was made by hand, though. And it was impossible, like it had to be manually synthesized. It needed to be filtered. It had iron filings in the reaction. It had to be filtered by hand. So what we did was we redesigned the starting material with a nitro precursor, and we put a protecting group on the indole and found that we could do a 2-step, 1-pot reaction, and it works really, really, really easy. We shared it for multi-center clinical trials, and in 2020, it became the first and only FDA-approved radiopharmaceutical for imaging Alzheimer's disease. It's been used in multi-center clinical trials worldwide. Well over 20,000 scans have been done, and it's used to de-risk drug development for Alzheimer's drugs right now. This method was patented and licensed to Eli Lilly, and again, this is something that we found worked really well in humans because these are those PIB scans showing the plaques. So what we're looking at is normal subject, amyloid positive, mild cognitive impairment, and Alzheimer's dementia. And you can see they're very different, and the PIB scans start spreading in through the cortex as you would expect from the neuropathology. So the PET scan is extremely sensitive for that. Shortly after that, I was contacted by Merck to work on their 2nd-generation Alzheimer's tracer for tau. And in that case, it was at the same stage. They had a lead, but the radiochemistry hadn't been worked out. So we optimized that and figured out how to label it for multi-center clinical trials. And again, you see the Alzheimer's subjects here versus the healthy elderly, and it's a huge difference. There's no question. It's something we manufacture multiple times a week at our site, and we ship it all across Ontario and into Quebec. We send this tracer to Pearson International Airport 4 days a week for multi-center trials. The reason, this is another area of using, it's now become required to use amyloid PET and tau PET when moving forward with these new drugs for monoclonal antibodies to reduce for Alzheimer's disease that were just released by Biogen, Eisai, and Eli Lilly. So again, PET has become central to this process. What we're imaging is tau, which is actually very complicated because tauopathies are characterized by these aggregated microtubule-associated protein and divided by 6 different isoforms and based on their repeat. So 3 repeat tauopathies are things like Pick's disease. 4 repeat tauopathies are things like CBD and PSP, often misdiagnosed for Parkinson's. Alzheimer's disease is a mixed 3R4R tauopathy, and another one that's really got a lot of press lately, particularly from American football and head injuries, is CTE, which is another mixed 3 and 4R tauopathy. For those of you who followed this area of chronic traumatic encephalopathy or CTE, the brain injuries are really the reasons. It's been known that getting people who get hit in the head, such as Muhammad Ali and boxers, they used to call it punch drunk in the 1920s. Yeah, these people would come in with symptoms because they'd been repetitively hit in the head. In 1937, they called it dementia pugilistica, and then in 1949, it became known as CTE. Bennett Omalu is a neuropathologist who was working in Pittsburgh at the time. And he started looking at postmortem brain tissues, particularly from American football players. And one of them, people might know, is Iron Mike Webster, who played for Pittsburgh Steelers. And he looked at his brain and he started seeing these tau tangles. So he's saying tau is definitely a biomarker for CTE. Interestingly, Will Smith played both Muhammad Ali and Bennett Omalu in movies. If you haven't seen Ali or Concussion, I recommend them. But if you haven't seen these Will Smith movies, I'm sure you've seen these Will Smith memes. All right, so we now get brains from the Boston University CTE Center. They send us NFL football player brains. We get the slices delivered to our labs in Toronto. And my PhD student at the time started testing all the different tau tracers in these tissues to see how does it look in CTE4. And what we found, the biggest thing we found is you can't use Alzheimer's tau tracers to image CTE because Alzheimer's tau is different than CTE tau. We've also tested the same thing in ALS. We've worked with different pharma companies like Takeda to try and tease this out. But the harsh thing is like, it's not Alzheimer's tau, totally different. We need specific agents. However, we can get some information from them. Here's a couple of examples where we do get some selective info. This is a former NFL player who is symptomatic of brain injuries, of traumatic brain injuries, and remember, CT is only diagnosed post-mortem. We do not have an in vivo way of doing it. Our goal is to image CTE in life. This is a hotspot in the gray matter. Sorry, it's called the cell side between the gray and the white matter. We've since partnered with the NHL, the CFL, and the Concussion Legacy Foundation of Canada and of the US, and Canadian National Defence to try and study military veterans and to study anyone that may have sustained a head injury. I'm personally and professionally interested in brain injuries and veterans' mental health. My mother served in the Canadian Armed Forces for over 30 years, actually here, starting at Hamilton Militia District. So that's where my family started here. And but a shocking statistic is that 2 US military veterans die every hour of non-natural causes, and one of those is a confirmed suicide. So can you imagine death by suicide at least once per hour, just in the US alone? And that would apply to any military, any country, anyone, anywhere that has a military. So the people we're now studying are military breachers. Most of the injuries actually happen during training exercises and not from people who've been deployed. You would assume there'd be more from people who are deployed, but it's the training. Military breachers are the ones that have to break into the buildings, and they practice this all the time, and they are exposed to shock waves constantly. We have this is unpublished data with a military vet who did not have any exposure to blast, and that's a breacher. So there's no question that there's a dramatic upregulation of tau protein in that brain. But it kind of has to be more extreme for the Alzheimer's ones to work for CTE. So our goal was we took a snapshot of all the different types of tracers that were developed for Alzheimer's tau by every company, including Roche, Genentech, Johnson & Johnson, Merck. But what we started focus, and a lot of those have had different FDA and European designations to be fast-tracked to look at non-Alzheimer's tauopathies just because there's nothing for them. So we started focusing on making PET tracers based on this pyridinal indole scaffold to optimize for non-Alzheimer's disease tauopathies, things like CTE or PSP. So we started with the more pure diseases, because so what we do is we took this compound called CBD2115. We were working with this colleagues who had spun out of AstraZeneca in Sweden, and they had this as a lead. So we fluorine 18 labeled it, and we also labeled it with tritium. It's called CBD2115. You can see this is human tissue, and these are KD values. So this is how, the lower the number, the more potent it is the easier way to think of it. And we're looking for like low nanomolar affinity. This is Alzheimer's disease brain. We also looked at 2 4R tauopathies, PSP and CBD, and it was quite potent in PSP. Pick's disease is a 3R tauopathy, and this is Parkinson's disease, which it didn't really bind in these. But it looked like a really good one for the PSP tau, so we said, let's label it with fluorine 18. And you see these black holes you see from rodent and non-human primate, means the tracer did not get into the brain. So despite all these hassles and headaches, and it's not easy to make these compounds and then finally get them in vivo, we get these black holes where it doesn't get into the brain. So we've written a review on how to get tracers to cross the blood-brain barrier. Just to fast forward really quickly, we use BBB scores, we use CNS MPO, these in silico methods, and we started to red flag things on this molecule, saying, okay, you know what, it's too many hydrogen bond donors, let's start making more compounds. So we just really hit this with a sledgehammer, made another 150 derivatives, and all of them had less hydrogen bond donors until we got this one called OXD2314, and you can see it's even more potent in PSP, more potent in CBD, and Pick's disease, which is a 3R tauopathy. It did not have any off-target binding. We were very excited about this tracer. So the next thing we did is we radiolabeled it, took it into rodents, and instead of seeing a black hole this time, the brain lit up like a light bulb, which was really exactly what we wanted to see. This is in non-human primate as well, and male and female, two species and sexes, and it got in the brain. We didn't see any troublesome radiometabolites, so we put a submission into Health Canada. And just 1 year ago, we translated it for first in human use. This is in a normal subject, tracers in the brain and clears from normal tissue. That's exactly what we wanted to see. And we showed that it was safe to scan. For those who are interested in the imaging side, and I'm really happy that we have a couple from nuclear medicine over here. We don't just go with the pretty picture, we now have to quantitate it. And this is where the physicist teams have to come in, and they say, okay, instead of pretty picture, give us numbers. Tell us how much is in all these different brain regions, show us time activity curves, do this different modeling. And on the chemistry side, we do column switching HPLC because we have to make sure that there's no lipophilic metabolite in the brain, because PET doesn't distinguish the form of the tracer. We have to make sure it didn't become a radiometabolite that we were imaging, and we had to show that it stayed stable over 2 hours. Now we've reduced the scan time. We've removed arterial, before we had to draw arterial blood, and we think we're going to be able to, we've already started now scanning people with suspected CTE and patients with PSP. We've also worked on other tracers, for example, following the patent literature. I think that's a really good way to try and make new tracers. So we followed the patent literature from this company, which is now bought by Lantheus, called Life Molecular Imaging. And we tracked down this lead called F4. So we radio-labeled it, we tritium-labeled it, we tested it in Alzheimer's disease, PSP, and CTE, and we could show baseline and block, which shows selective binding and staining. And then we actually contacted I showed this data to the company. And we were at a conference, and they didn't say anything. And then a week later, he said, I think that's the same tracer that we're working on. And he said, we're going to take that to human. Why don't we just join? So we said, well, why don't we join forces? Rather than us publish something, you publish something. So they took it into human. It has slight uptake in the skull. So fluorine sometimes falls off and has bone uptake. So it landed in the skull. Otherwise, it was actually a very good tracer, and that could probably be modeled out. But if we didn't have the OXD tracer, this would have actually been a very reasonable alternative to their lead compound, which is shown below in Alzheimer's disease. Now, Alzheimer's disease is a complicated disease. It's very difficult to drug, therefore it's very difficult to image. There's many hypotheses, many drugs, many targets. So in imaging and biomarkers, we look at A for amyloid, T for tau, N for neurodegeneration, which is usually with FDG. And I've shown we have tracers for all 3 of those. But then over the last few years, I think during COVID, people had time to write stuff. So the person who came up with ATN decided he was going to expand it. He said, no, it's more than that. There's I. I'm adding I for inflammation. And we're going to look at things like microglia and astrocytes and start looking at different mechanisms in inflammatory pathways, because these have to be relevant in Alzheimer's disease, and they're definitely relevant in CTE. Okay, so these are all the different types of tracers that we've explored, like from cannabinoid targets, monoamine oxidase A and B, many different cyclooxygenases. So I'm just going to show a couple of quick examples of things that we've done in inflammation. So I feel CAMH has sort of been at the forefront of brain inflammation because going back to 2008, we made this compound called FEPA. It hits something called target called TSPO. It's not specific for, it's more specific for microglia, but it's not an ideal tracer, but it was a second generation compound. We went from rodent, where we were studying tumor xenografts, all the way to first in human. But what's cool is we shared it with a bunch of other labs, and we collaborated with laboratories up in Long Island, for example, Stony Brook University, and they ran this study where they brought in World Trade Center first responders and scanned them with FEPA. These are the people who showed up during 9/11, and most of them had symptoms that were very much like people who'd been deployed to combat. They had PTSD, they had been exposed to neurotoxins and gases, a lot of them had head injuries from the process, so there was an increased uptake in the World Trade Center first exposures. And just a couple of months ago, this paper came out from a collaboration with UCSF, where they were looking in San Francisco at people with HIV. And they compared smokers with HIV to non-smokers and found that actually smoking was neuroprotective, interestingly. So that was a cool finding for another reason, because nicotinic drugs could potentially be a viable option here. So it's kind of neat that they found that. Anyways, cool studies that have come out. Those were a couple of snapshots. Another thing I think we need to be doing in our field, and people who are looking at brain imaging, is starting to work with people who study oncology, because I don't think anyone understands pathways better than oncologists. So in neuro, we're very good at looking at this receptor ligand interaction at the surface of the cell. We can image transporters, enzymes, anything like that. With large molecules like antibodies, affibodies, and peptides, again, we're still at that extracellular domain. But what we're interested in is following what the pharma companies are doing and saying, what's going on inside the cell? Can we look at signal transduction cascades? Can we look at kinases, things like that? So one open science project that we've now done to look at a kinase is called activin receptor-like kinase 2 for a rare orphan disease called DIPG. This one is extremely sad. It's diagnosed in children between 5 and 9, and they all die. It's really, really sad. There's nobody survives this. It's got pretty much a 0% survival rate. It starts in the pons, which is this region of the brain. And the Ontario Institute for Cancer Research was making drugs for this target with medicine for kids pharma. And what's really cool is that my postdoc collaborated with a former postdoc of mine who was at OICR, said, let's do an open science initiative for this, get all our papers out there, and can we join forces with them and try and come up with something for target engagement? So can we label their compound? So we labeled their drug, but we got a black hole, because again, a good drug does not mean it's going to be a good tracer. We're looking for very different properties when we're using PET tracers. We're looking for something that gets in the brain and clears and hits the target. So my postdoc at the time made several derivatives and carbon-11 compounds until things got into the brain. And now we're still refining that right now. We were recently funded by the CPDC. I'm not sure if Owen Roberts was here, but he was actually part of M4K Pharma before he became CEO. So he's been involved in the project with us for the whole time. And we've had really good support and collaboration with them as well. We're also interested in taking drugs for cancer and applying them to the brain, because if there are, or radio tracers that are already there and validated, there's no reason you can't repurpose them. But we're doing it now in reverse. We think that our neuro-PET tracers could potentially be used for cancers. And examples of this would be like cyclooxygenase 1 and 2 inhibitors, which could be reapplied for oncology and women's health. Right? So we took two tracers from the NIMH called PS13 and MC1 and applied these to ovarian cancer xenografts showing baseline and block. And again, with this one, we used it for colorectal cancer using baseline and block. This was done by, she was a postdoc in the time, Amy Boyle, and she's now a faculty member at our center, looking at links between cancer and brain and trying to learn and use the activity vice versa. I thought I'd tell this story because I started, after my postdoc, I had to come up with a project for, you show up in a lab and we didn't really have any startup, and I was just reading the literature and I saw these really cool Nature papers on a target called glycogen synthase kinase 3 or GSK3. And this is the compound that they had from AstraZeneca, and I looked at that and I said, oh, that's a piece of cake, I can label that. I can synthesize that, and I did it the same day. I said, all I have to do is demethylate it and put a carbon 11 on it. That did not work out well. It's a long, long story, but about 8 months later, I ended up making it completely different route, multi-steps, terrible nightmares about this. But the point is, it's a breakthrough in antidepressant development. It has applications in oncology. I thought this was a perfect project for me because if I made a tracer for this holy grail target, we could really work and get new antidepressants. And then there's the GSK3 hypothesis of Alzheimer's disease. Went through all that hassle. And again, I got the black hole for the brain. The tracer didn't get in. So I made all this work to synthesize the compound, synthesize the precursor, carbon 11 label it, get it into rodents, and it totally crashed and burned. Because that's FDG for comparison, which lights up like a light bulb. So I think for anyone starting, I want, and also anyone working with radiochemists, be nice to radiochemists. There's many places we can fail. We can fail in the chemistry. We might not get a potent and selective compound in the med chem. We might not be able to radiolabel it. And then even after all that hassle, we can fail in the imaging. And that's where we mostly fail. So I think there are two things we can learn. One of them, publish the basic science. Don't just bury it saying this is boring, I don't care about it. Just get it out there because you don't know. And the other thing is we need a high throughput approach. We can't be making one tracer at a time. Like this was a year of my life to get a tracer that didn't get in the brain. And I kept doing this. And then I became the world's expert at failed radio tracers for GSK3. And I was at a conference one time talking about this. Interestingly, I get at the coffee break, some guy taps me on the shoulder and said, I'm the GSK3 lead from Pfizer. You can come crack our library. We have millions of compounds. It was like winning the lottery. So we went through his database. He came over to our site. He brought this proprietary database. We searched structures for what we wanted for PET, not necessarily drugs. Potent selective compounds, things we could methylate very easily. And now we had tracers that got into the brain and that we could block. So this was just game-changing for us. So again, that led to many, many grants, many, many papers, things like that. We've looked at sex differences. We've looked at it in cancer. We've done a lot. We've even made second-generation tracers, taking it from low brain penetration to high brain penetration. Now we even have a fluorine-18 analog, and we're planning to translate that to humans in our collaboration with the Yale University PET Center. The second thing is we need library designs. We need to make compounds much more efficiently. In pharma companies, they do combinatorial chemistry. Technically, they really make massive compounds that are linked by carbonyls. So we have a colleague at AstraZeneca and asked him, can you look at your central nervous system database, tell us how many compounds have methyl groups on them? And he scrolled this massive database. He said about 15%. And I said, how many have carbonyls? And it was about 80%. So you can imagine that how much easier it is if we can label carbonyl groups. So inspired by green chemistry and trapping CO2, we normally take CO2 from the cyclotron and we convert it to methyl iodide. That takes an extra step. So we said, let's just trap CO2 in a fixating base like BIMR DBU. And it worked perfect. We just quantitative trapping. And then you can throw the left half of a molecule and right half of a molecule in, and you can make libraries of compounds. And I'll show you a few examples of how we can make libraries of carboxylic acids, oxazolinones, ureas, or carbamates. Oops, what happened there? Okay. Here's an example for monoamine oxidase B. This is the simplest reaction that I did, and I love this reaction because we didn't bother with even putting a left and right half of a molecule. We just has an amino alcohol, CO2 just snapped in, trapped it in a fixating base, just bubbled it in, and dehydrated it with POCl3, so that just pulled out the water, and you have the 5-membered ring. The reaction is so simple. It's done at room temperature. There's no tricks. It could be literally done on this podium behind a couple bricks of lead. It's so easy to do. So we've taken that tracer into human for monoamine oxidase B, and that's an anti-astrocytic marker. We've even used it in traumatic brain injuries, and you can see the difference compared to healthy control. There's a higher upregulation. Here's another example of how we use carbon dioxide fixation, again, for human use. It's not just an academic exercise. You can throw in any amine and any phenol, and any of us can go through the Aldrich catalog, and these are the most dime a dozen compounds. These are the cheapest compounds you can buy, and literally throw them on with one standard method. It's so simple. And like I said, you can vary anything on this side and anything on this side. So the other reason it was critical to label at the carbonyl group in this specific case, this was for a cannabinoid receptor path compound. The enzyme is called Fa, or fatty acid amide hydrolase. What happens is Fa shows up and it binds here and it bumps off the aromatic ring, and that would be the logical place to label this compound with carbon-11 or fluorine-18. So this labeling was critical for staying bound to the enzyme. We took this tracer into human. This is baseline, and then blocking means the patient or the healthy control took Pfizer's drug, and you can see 30 minutes before the scan, and you can see it blocked it. That means it's definitely specific. We've also more recently looked at healthy controls and cannabis users, and you can see cannabis blocks Fa. So it definitely has a CNS impact. Another nice thing about this is it's library synthesis, so radiosynthesis, so you can make many, many compounds at the same time. And it led us to look at other compounds in the cannabinoid pathway. We made one for a target called MAGL and one for CB1 receptors, cannabinoid receptors 1. Both of those are planned for human use, first in human use this year. The ATN criteria got expanded again. I think, again, people had too much time on their hands during the pandemic. And they said S for synaptic density. So synaptic density is very a new thing to image. Again, big problem is you have to label non-activated aromatic rings for the leads. But we collaborated with the Yale PET Center because they were working on synaptic density PET imaging agents. And we got a grant through Michael J. Fox Foundation and the Western Brain Institute to work together. We weren't sure which tracer to move forward with, so Yale supplied us with a couple of these precursors. And it worked pretty well to make this compound called Synvest. Again, this is a copper catalyzed reaction with a tin precursor, and we do this routinely for human use. Again, we have to do a lot of extra testing because it's metal catalyzed. But it's one way to get fluorine onto the aromatic ring. What was troublesome was when we started going to human studies, our scans did not look like Yale scans. And then we had to check the supplier. They sent us GMP material from the site in Germany. And it turned out that the chemist ordered the wrong material and released the product. Instead of buying the 3-methylpyridinyl compound, they bought the 4-methylpyridinyl compound. So nitrogen was in the wrong position. And this is an example, interesting, because this is an example of the fluorine effect. The fluorine effect is when fluorine on a molecule in a different position has dramatically different biological behavior. And we saw that with fluorodopa, and we see it here with this as well. We've now cleaned that up. Fortunately, they recalled that product, and it was something you couldn't catch by the characterization. Even just the standard proton NMR and mass spec, everything would match. So one of our colleagues ran a 2D NMR massive study to show that they actually had the nitrogen in the wrong spot. And then they had to recall it worldwide. We've done over 300 scans with that tracer alone. If any grad students or anyone in the room is interested in making $2 million, the Michael J. Fox Foundation has a prize for the first lab to make a PET tracer for alpha-synuclein for Parkinson's disease and make it publicly available. I brought this up at one of my group meetings. One of my postdocs was like, I'm in, let's do it. So she started making all these compounds. She made well over 50, and we labeled this one called ASIN-44. It looked great. It got in the brain, it cleared. It's exactly what we wanted to see. It super potent, everything was great. We were like, all right, she's gonna be cashing this check any day, but we always have to double check that it doesn't have troublesome brain penetrating radiometabolites. And unfortunately, this tracer metabolizes in the brain. So again, we were stuck with another failed radio tracer for alpha-synuclein like everyone else in the world. And we were just like, let's just publish this. No one's gonna read this. I had an invitation for Kemur J, just get it out there. Oddly, as soon as it went out, I got contacted by 3 academic universities and a pharma company. So Takeda, UPenn, UPitt, and 2 in Europe, including Tübingen and another site in Italy and University of Copenhagen. So multiple sites wanted to work with us on this because they said, this is potent. Let's join forces and work together. So now they said, let's see if it's a problem in higher species. Let's make more derivatives. Let's compare it to the gold standard. So we've got this whole new collaboration with a bunch of sites that we would never have thought we'd ever work with. So again, publish the negative data. Coming back to labeling non-activated aromatic rings, this is definitely a thorn in the side in the community, but it's also one of the most exciting areas of research. After you saw that detonation that I showed, which blew out the fume hood in Professor Strobeljan's lab, I said, no need to make fluoride behave like F2. But my students kept bringing all these amazing papers from all over the field trying to make fluoride go onto a non-activated aromatic ring. So these are literally bullet points from one of my lab meetings. we said, okay, if we're also going to do this, these are the check boxes we have to have. Our method should be metal-free, give high molar activity, be shelf-stable. It should be one-step, regioselective, and have wide substrate scope. And my extra criteria is I want to actually use it in humans for drug development and first-in-human PET studies. If we're not doing that, there's no point wasting our time going on such an effort when every other organic lab is doing this with way more resources than us. So we started working with these hypervalent iodine precursors, iodonium ilids, and we got these rock-stable crystals. It was really nice, and they seemed really good, so we published the academic paper. And then the first thing we had to do was solve a problem in the lab. That's what I wanted to do. We were making this compound called FPEB multiple times a day for the Harvard Aging Brain Study because PIB shows the amyloid, FDGs, glucose metabolism, functional MRI is good for glucose hyperactivity. But PEB has an advantage is that in early Alzheimer's disease, whereas everything else is degenerative and the signal goes down, this one goes up. So we needed to make this. And while it looks like such a simple reaction on paper, it's actually a nightmare because you have the nitrile meta to the nitro group. So fluoride does not want to go there. It's less than 1% radiochemical yield, so we had to start with a huge amount of radioactivity every time to make it. So we used iodonium ylide chemistry, and it was game-changing. It went from 1% to 1 to 2% to 20% radiochemical yield. We've shared this method with many academic sites, including Yale, VU Amsterdam, Beijing Normal University, that are all using it for their human use PET studies, different tracers, different problems, and 3 pharma companies are using it, including Merck, Pfizer, and AstraZeneca, for human PET imaging for their drug programs as well. So that we really like that method. It's when it works, it really works well. Here's an example of when Pfizer contacted us after that paper came out and said, can you help us de-risk this study? It's a tracer for non-small cell lung cancer. This is a 2nd generation compound called lorlatinib. Crizotinib was the 1st generation. And they said, we want, none of those compounds get in the brain, but we want to see if our drug gets in the brain so it can actually hit the brain metastases. Can you label the compound and show us our new generation compound gets in the brain using PET? So we started with carbon-11 and we showed it did. You can see it's in the brain. And then they said, okay, now for multi-center trials, we need F18 on it. And that's not trivial. It's very unproductive SNAR. It does not work with a traditional nitro precursor. So we used iodonium ylide chemistry and it worked really, really efficiently. We get about 20% radiochemical yields, really painless, and again, no tricks involved. Another application of iodonium ylide chemistry that I really like is here, because when we published a ChemSci paper, we needed an example of a compound that could be made. So we said MIBG is a guanidine derivative labeled with iodine 123 for SPECT imaging for children with neuroblastoma. Like now, unlike the neuroblastoma, unlike the brain cancer I showed you before, DIPG, where all the children have a very poor prognosis, if in neuroblastoma, if you catch it early, 95% of the children survive. So it's critical to catch this early. But this is the resolution of the gold standard, MIBG. It's terrible compared to PET. This is called MFBG. And we used iodonium ylide chemistry to make this, and these labs in Amsterdam and across the Netherlands and now elsewhere ran with it. And they started making this with our iodonium ylide method. And the really nice thing here is compared to the SPECT method, it takes 90 minutes. You have 2 visits, thyroid protection. This is really hard on kids, and limited resolution. Whereas PET, you get high resolution, 10-minute scan times, no anesthesia, no thyroid protection, 1 visit, and it's a lower dose. So this is something that I think will hopefully take off a lot more, and many labs around the world are using that. Kind of one of the last projects that I wanted to talk about for a new chemistry point of view is like bringing my life full circle. When I was a postdoc, I was making radio tracers for this cancer target called EGFR tyrosine kinase, and everything had an acrylamide on it. And I hate those because they are microacceptors. They react with everything. They polymerize. They're terrible to work with. And the only way you can make them is from Grignard reactions. It's almost impossible to extrude CO2 from the atmosphere. So instead of doing that, I did it with fluorine 18 in 10 steps, which is extremely inefficient, but I basically synthesized the molecule almost the same way I made it in the cold lab. I made fluoroaniline in about 5 steps, and the intramolecular cyclization to form the ring, make a quinazoline, and then I'd reduce the nitro group and then acylate there. So Again, not easy, not trivial, but it shows you can push fluorine 18 out to 10 steps. But then we started working with Sanofi, and they asked us to make another acrylamide. And I was like, oh my God, I can't do this again. So we bought a carbon monoxide system and took CO2, converted it to carbon monoxide. This has been used quite a bit, but no tracers had really been translated to human use with this method at the time. So we found that we could do these reactions, again, with palladium and nickel catalyzed reactions to make it directly from CO2. And again, this was another paper. In the first week of the pandemic, I thought I'd be back at work every week for the next 2 years. But first thing I did was my postdoc had gone back to Sweden. I contacted the guy at Sanofi and he said, well, we're not going to run that PET study that you set up for us anymore because it's COVID. We're just going to run the trial. I said, well, then can we just publish the paper? He said, I don't see why not. So I wrote it up thinking no one's going to read this. Again, invited issue of Journal of Labeled Compounds and Radiopharmaceuticals. Impact factor might be 1 or less. And I said, okay. But oddly, the paper goes out and I get contacted by Bristol Myers Squibb saying, can you help us with this? Because we have BTK inhibitors as well. So we've joined forces with them. And now we've helped them de-risk their drug development program and moving their tracers to human use using CO chemistry to make acrylamides. The last thing I just wanted to mention is there is a major need to train radiopharmaceutical scientists. Right before the pandemic, again, 2 weeks before, I was at a meeting for the IAEA, the International Atomic Energy Agency in outside of Vienna. And they, one of the things we said was in our focus group is there's a shortage of radiopharmaceutical chemists. There always has been. There was since I was a grad student till now. And it hasn't been filled. And it's hard to, and radiopharmaceutical scientists are scarce because you need to find people with chemistry, biology, mathematics, physics, engineering, medicine, radiology, everything. So it's a huge field. So I thought rather than, like, we wrote a review on, we have to do this, but then I was like, well, what's the point of writing that review if we're not going to do anything about it? So I wrote a MyTax grant, and we got the largest one ever funded outside of NSERC. And it can train 150 years of graduate students and postdocs over the next 2 to 5 years in radiopharmaceutical scientists, but it's contingent on industry-sponsored agreements. So it's a matching fund program. So it's something I'd love to see if anyone was interested in partnering on. If we could take advantage of this MyTax, that would be amazing. Just in conclusion, Canada is a leader in brain PET imaging. New tau tracers have gone to human use. I've shown many new radiochemistry methods are needed to expand the tracer toolbox, so we're not restricted in our methods. I also think these new scanner technologies are really going to shape the field because they're becoming extremely sensitive now. I also think there's a lot of opportunities for academic and pharma partnerships. I think pharma companies are very happy to work with academia, which may not have been the case before. And I think we do need to train more Canadians in radiopharmaceutical sciences. I hope the MyTax will help catalyze that with the partners. Again, just want to thank all the support we've had from our funding agencies and pharma partners and people along the way. And again, massive thanks to Sam, Alex, and the Brockhouse lecture organizers, McMaster faculty, staff, and students for all your kind hospitality on my visit. Past and present mentors of particularly Professor Strobeljan and Dr. Helen Mercier are both here today, who are very much part of my whole academic journey. And I really do owe my everything in my career to my training at Mac. This has been absolutely fantastic. The chemistry program here was brutal and rigorous. It was not easy by any means at any step, and I really do appreciate that. It was, and also so many past lab members and colleagues, like for example, Mark Bordelis and Karen Stevenson. I can see many more in the audience too, Sahil and others. And it's just Rezwan, so many people that I've been fortunate to work with. And sorry if I missed anybody, but really appreciate all the collegiality, the mentorship, and thank you all for your kind attention. It's been an honor to speak here today.
What a force in nature, seriously. I don't know if you guys appreciated that, but starting with something like clinical need, basic science, chemistry, resourcefulness, connecting the dots, then doing clinical translation. This guy just cannot be stopped. I was talking to him a little earlier. He tried to get me to a PET scanner. veterans in the military. I'm like, I don't look at my brain. I don't have my next brain. I don't. That's the speed.
I didn't know about your military training background. That's so cool. We'll bring you in.
But beautiful.
I'll start. Beautiful talk, amazing,
Okay. Sorry.
amazing. And I have lots of questions about various chemistry, but a more broad question that I have, sort of thinking about the way people are doing work today, moving into the future. One thing that you didn't talk about is machine learning. And we have a lot of, like, there are all these libraries of molecules, and there's a lot of data that you have collected on all these different compounds. It seems like it would be right for using machine learning to try to pinpoint specific aspects of molecules with great brain or find certain things. Is that something that you're
The hard part for PET is we just don't have the large volume of compounds that they have in drug development where you can really screen a massive data set. There are people doing this though. There's a group between Michigan and Stanford that are doing it specifically for PET and looking for correlations and trying to predict what's going to work. Our experiences though, really it has to be just, we have to label the compounds and get them in vivo, just because if we don't do that, we get very surprising results. Sometimes you can really have the most amazing in silico prediction, but it just doesn't work. But what AI can really do for us that I think is going to be super helpful is in the radiology reads. Because if it could go through a massive data set of all the people who've had, for example, FDG scans or different scans, even new tracers, and look for patterns that could get missed by a visual read, and then say, okay, wow, you know what, from this subset, we thought this was Parkinson's, but it's PSP because we know what to look for, or it's something else that's misdiagnosed as Parkinson's, for example. In the Alzheimer's disease spectrum, that would be huge because it's Alzheimer's disease related dementias. There's so many things that I think AI would be able to say, okay, from the MRI and the PET scan, this is a pattern we're noticing. And the patterns and going back through that database would be, I think that can be game changing for us.
That's great. Well, Alex asked a very general question. I'll ask a really specific one, I guess.
Sure.
Yeah, you provided many fine examples of the challenges associated with labeling unactivated C6 rings, I'm very sympathetic in that regard as well. But I was just wondering, you did mention MK or
6240.
that described chemistry there, but one thing that did surprise me, it doesn't surprise me that you got enough for bright scans, but you mentioned that you ship it out, and that does contain a Did you use iodine in that case?
No. I can I can show you that slide. We can pull it if that's easier. Just give me 1 second.
And just as a follow-up, how much do you need to make in order to get it to your location?
Okay, so how much do we need to make would be, sorry, just give me 1 second. This is what you're looking at, right? Oh, it's not switching. While it looks like a non-activated aromatic ring, it's got a pyridine in the second ring. That was the reason it's I believe it's more, and it has the amine next to it as well. So it's not quite not activated. It is activated enough to take a fluoride displacement. We get 20% radiochemical yield consistently, which is really good, but actual isolated, yeah, in hand. So we make it, and that's what we ship multiple times a week. So yeah, it's a very good reaction, and we can start. We have a high energy cyclotron, 17 MeV, so we can start with 2 curies, and we make enough, ship out, quite a bit, so they can do multiple scans with it. We even ship it to Montreal. Yeah. Yeah, I can show you that.
That's okay.
Okay. It had an aromatic union in that, and it had an amine next to it, which helped. So it worked. We figured it out. We tested it cold first. We did that model compound, then we just went straight to the main compound.